Hello guys, welcome to my channel. So as you know, in the previous video we studied about the DNA replication process, right? How the DNA polymerase is used for the replication of DNA, but there is more to this replication process. So this is just a basic thing that we studied in the previous video. We'll go through one or more things right now in this video. So as we know, DNA polymerase is used as a replication for, if for example this is the new strand in the DNA polymerase adds a new DNA or new nucleotide rather to the new becoming strand. But the DNA polymerase itself cannot start a new DNA replication at the point at the replication fork. So all right, I will remove this and will show you. So what will happen? So right, so for example, as we have here, so we have the two strand. If this the replication fork is going on this way, right, we have a DNA polymerase. But before the DNA polymerase act, right, so DNA polymerase needs something to start the replication processes, cannot start itself. So the help that the DNA is getting is by primase enzyme. So this enzyme primase is an example. So this primase is an example of RNA polymerase. Right, so why RNA is used as a primase for the replication of DNA? So this RNA, ribonucleic acid, right? So DNA is deoxyribose nucleic acid. So the difference between DNA and RNA is the sugar, and there is a base as well. So we'll discuss that as well. But so this RNA, it makes a short length of closely related type of nucleic acid, that is the RNA. So this, what it does, it makes a short length of just maximum maybe 10 nucleotide long and is a base pair to the template strand. So that 10 nucleotide will be base pair to this the template that is being used for the replication process, right? And thus provide, so what it does, it provides a 3 end. As it is similar to DNA, it provides a 3 end as a starting point for the DNA polymerase to begin the replication process, right? We know we have a 3 end and a 5 end, right? So this is 3 and 5, right? So this is our template strand, and the new one is made from this to this side, right? So why 3 end? So as this is a 5, so if just one nucleotide, for example, I'm just drawing a square. If this is a nucleotide with a base, for example, C, so it should have a 5 end. So this will be 3 end. So if this is RNA, so it will join here one more, right? Maybe this is A, for example, and 5 and 3, right? So this provides the 3 end to the DNA for the process of replication to start. And the enzyme that synthesizes the RNA primer is called primase. So that's why it's known as primase. And the primase is an example of RNA polymerase. So this enzyme synthesis RNA using the DNA as a template, right? So a strand of RNA is very similar chemically to a single strand of DNA, except, right, as I mentioned, so RNA is ribonucleic acid, DNA is deoxyribose Nucleic acid. So the difference is, first, the sugar, DNA has the deoxyribose, and RNA has just a ribose sugar. And RNA and DNA also differs in the base pairing. So this base group that it has. So only in ones though. So like DNA, we know it contains thymine, right? T. DNA contains thymine as a base, right? But RNA does not contain thymine. It contains uracil. Yes, so that's the only difference. But why RNA is still used? Because the uracil can form a base pair with the adenine of the DNA, and the RNA primer is synthesized on a DNA strand by complementary base pairing exactly the same way as a DNA does. That's why RNA is used as a primer. Right, so what it does, so for in the last video we studied leading strand and the lagging strand of the application process, right? So for the leading strand, what happens, so the RNA primer is needed only to start the replication at the replication of origin. For example, as we discussed, like for example, this is our replication of origin, and here the replication fork will start forming. So once the replication fork start forming like this, so the at this leading strand, so the RNA is just needed at the start of the replication fork, at the replication origin. So what it does at the leading strand, so RNA will put two of it nucleotide here to join 1 and 2, for example 1 and 2 nucleotide to join on this template strand, and then it will provide the DNA polymerase with the 3 end. So DNA polymerase will start adding here, and the leading strand will keep on moving. But for the lagging strand, as we discussed, it is done by the Okazaki fragment, right? This lagging strand is done by the Okazaki fragment. So what the RNA primer do here on the lagging strand, so here the DNA synthesis is discontinuous, right? It's not continuous segment, because like here the Okazaki fragment, like 4 or 5 will join, and then it will join the template, then again 4 or 5, and then it will join the template strand. So this is the discontinuous DNA synthesis. So new primers are needed to keep polymerization going. Here just like couple of them are needed for the leading strand, but for the lagging strand there are always a new primer needed to keep the lagging strand for moving on the polymerization. So the moment of replication fork continue, so as the replication fork moves, so it continually exposes, right, unpaired bases on the lagging strand here, on the lagging strand, and the new RNA primers are laid down at an intervals along the newly exposed. So I will show you. Right, so for example this, right, so this is our lagging strand, for example, and this is our leading strand, right? So we'll put the lagging strand separately, just we'll use this as directly as like this, and we'll name as lagging strand. All right, so on this lagging strand, what happens, so the RNA primers are laid down at intervals. So for example, this, as the replication fork is moving, it is opening the unpaired bases. So the RNA primers, for example, this, it dropped RNA primer here, then the replication fork is still moving. As soon as the new one unpaired bases come, so it drops here. So the RNA primers are laid down at intervals along the newly exposed single-stranded stretch. So what the DNA polymerase will do? DNA polymerase will add the deoxyribonucleic acid at the three end of each primer. So like this is the three end of each primer. It will add a three end of the each primer to start a new Okazaki fragment, and it will continue to elongate. So let me make a space. Right. So for example, this is here and this is here. There are two strands. So the Okazaki fragment will start adding here, and it will continue to go until it hits the next RNA primer, and this is how the DNA polymerization goes on the lagging strand. Alright, so to produce this continuous DNA chain from many DNA fragments, right? So there is a DNA fragment here, there is a DNA fragment here. So there is a special DNA repair system that act quickly to erase the old RNA. So there is a repair system. So that act on, for example, this RNA and the old RNA primer and replaces it with DNA, right? An enzyme that is called DNA ligase. DNA ligase. So this helps to join the three end, right? You can see the three end of the new DNA fragment to the five end of the previous one. So this helps. All right, let me see. So this is here 5, right? So this helps to join this 3 to 5. It's done by how these two separate are joined together to become a continuous DNA strand is by the help of DNA ligase. That enzymes helps to join and to complete the process, right? So why might an erasable RNA primer is preferred to a DNA primer, right? That would not need to be erased, right? So why instead of like this RNA primer is used instead of DNA primer on the lagging strand as well? Why? Because so there is an argument that a self-correcting polymerase cannot start chain de novo. So self-correcting is the DNA polymerase is a self-correcting, and it cannot start a new chain. Implies why? Because it also implies the converse, like an enzyme that starts a chain anew cannot be sufficient to do a self-correction. So thus any enzyme that primes the new synthesis. So for example, as a RNA primer that primes the new synthesis of Okazaki fragments will be of necessity make a relatively inaccurate copies, as I mentioned that the polymerase that is starting a new chain cannot do self-correction. So as RNA is starting the new Okazaki segments, so it cannot do self-correction and it has relatively inaccurate copies, at least one error in 10 raised to 5. For example, this is an error ratio. So even if the copies retained in the final product, for example, if RNA is just used and if these copies are retained in the final product, constitute as little as 5% of total genome. That's it, not more than that. For example, 10 nucleotides per 200 nucleotide DNA fragments. But the resulting increase in overall mutation rate would be enormous. So therefore, it seems likely that the use of RNA rather than DNA for printing brings a powerful advantage to the cell. Right, the ribonucleotides in the primer automatically mark this sequence as a suspect copy to be efficiently removed and replaced. Alright, so this was the RNA first. So in the DNA replication process, as soon as the replication fork opens, so first the RNA polymerase is useful in starting the DNA replication, right? In the leading strand, it just need one or two nucleotide, but in the lagging strand, it will need the primase is needed continuously on the lagging strand because of the discontinuous movement or discontinuous development of DNA on the lagging strand, right? So RNA polymerase will start, and so on the especially on the lagging strand, when there are like RNA polymerase that are used as an Okazaki fragment, and when the DNA polymerases are coming to join it, so this RNA polymerase are removed by a repair polymerase. That's a kind of DNA polymerase, so that will remove quickly this RNA polymerase and it will be replaced by DNA polymerase, and then these two separate DNAs are joined together by DNA ligase, and this is how it is continuous. All right. Okay, so that was the DNA replication, but how the process start with the RNA polymerase. So we'll continue with the next video. So that's it for today video, and the next video we'll study the replication machine, the entire replication machine that is used, like what is used to open the replication fork, then the DNA polymerase, and there are different kinds of proteins that are used for the movement of DNA polymerase. So we'll study in the next video. Right, thank you guys for watching, and don't forget to subscribe.